It's 10:03 now. Let's get started. So, hi everyone. Welcome to today's seminar. So, I would like to firstly begin by acknowledging the Wurundjeri people of the Kulin Nation as the traditional owner of the land on which our Parkview campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining us from today. So I pay my respect to the elders, past, present, and emerging, and to the Aboriginal elders and the other communities. So my name is Hai Tian. I'm a CCEMMP student at WeHi, and today I'll be chairing this session. So firstly, let's quickly go through some housekeeping rules. So this meeting will be recorded and made available online at the CCEMMP website. Please stay muted while our guest speakers are talking. If you have any questions, feel free to ask it, or you can type it at any time in the chat or raise your hand, and at the very end of the session, there will be a Q&A session at the end. So today it's our honor to have Assistant Professor Oliver Clarke. Oliver Clarke is an Assistant Professor of Physiology and Cellular Biophysics at Columbia University. He finished his PhD at the University of Melbourne back in 2011, supervised by Jackie Galvis and Brent Smith at WeHi, where he found a passion for membrane proteins structural biology. He joined a group of Wen Henriksen as a postdoc from 2012 to 2017, where he applied a single particle Cryo-EM to understand the architecture and gating of mammalian cells' random receptor calcium release channel. In late 2017, he started his lab at Columbia University, focusing on the molecular architecture of membrane protein complex purified from native tissues. So I'll hand over to Oliver.
Okay. Thank you very much, Hai-Chan, and thank you very much to everyone from CCMP for having me here to talk today. Let me just share my screen. It is really great to be talking in Australia. I'd love to be there in person. Unfortunately, can't do that right now, but hopefully sometime in the not too distant future, I'll be back in person. Okay, so what I want to tell you about today is kind of a new direction for the lab. And it's really focused on understanding, let's say, protein communities, and with a specific focus on the erythrocyte ankyrin-1 complex. So what do I mean by protein communities? So just take a look at this illustration, this beautiful painting by David Goodsell. This is from, I believe, 2019 or 2020. And this is intended to illustrate the erythrocyte cell membrane as we imagine it to be. And, you know, there's a lot going on here, but what I want you to take away is there's a very crowded place, and that it's not just a simple, lipid membrane with proteins floating about. The architecture of the membrane itself is intrinsically interwoven with the cytoskeleton and with not just individual membrane proteins, but clusters of functionally related membrane proteins. And that's something that we wanted to investigate using a structural approach. So red blood cells, obviously the most abundant cells in the blood, and they play a critical role in the transport of respiratory gases, taking oxygen from the lungs to the tissues, and then taking carbon dioxide to the lungs where it's excreted. And you know, the most prominent thing about erythrocytes is that they have this characteristic biconcave disc shape, which both gives them a very high surface area to volume ratio, facilitating very fast gas exchange. And it allows flexibility to traverse very small blood vessels. And they do have a particularly characteristically strong membrane and flexible membrane for that purpose. And the shape and the flexibility of the RBC membrane is provided by the spectrin-actin cytoskeleton, which is a kind of hexagonal lattice of spectrin and actin that is tethered to the lipid bilayer by these spring-like ankyrin proteins, which are going to be a focus of my talk. Right, so what does that look like? So over on the right here, you can see a beautiful negative stain image from the early days, from the 1980s, that gives you an idea of what's left if you take a red blood cell and you solubilize the membrane. you're left with the skeleton, the ghost of the red blood cell, everything except for the lipids, more or less. And if you put that down on a negative stain grid, you see this hexagonal lattice beautifully. But what you also see is you see these complexes. So the edges here are formed by spectrin tetramers, so tetramers of the cytoskeletal protein spectrum. At the center, at this node, you have what we call the junctional complex, and there's one of those at each vertex, at each vertex, basically, of this skeleton. And then in the middle, you have another complex, the ankyrin complex. And this complex acts to attach the spectrin cytoskeleton to the membrane via, on the one side, directly interacting with spectrin using a spectrin binding module of ankyrin. And on the other side, the membrane binding domain of ankyrin mediates clustering of a bunch of different functionally related integral membrane proteins, the most prominent of which is the band-3 anion exchanger. And there's a lot of beautiful biochemistry from the 80s, 90s, and more recently, identifying some of these components and trying to identify, how they fit together, which I don't have time to go into in detail today, but it formed the foundation for all the work that I'm about to tell you. Okay, the one thing that I really want to emphasize here is that we're looking at this in the context of erythrocytes, but this is not an erythrocyte-specific arrangement in broad terms. So what do I mean by that? What I mean is that in other cell types, in other tissues, we also have spectrin-actin cytoskeletons, and we also have, ankyrin complexes, different ankyrin complexes. So for example, in neurons, and particularly in the axon initial segment and the nodes of Ranvier, we have these kind of circular arrangements of spectrin that go around the axon. And attached to those are ankyrin complexes that form clusters of sodium channels and potassium channels. And similarly, in cardiomyocytes, we have ankyrin complexes of sodium channels as well. So this is something that, while we're looking at it in the context of erythrocytes, because essentially that's the easy place to do it to start with, I think the implications and hopefully some of the themes that we draw out of this may have broader relevance. Okay, so as I mentioned, the ankyrin-1 complex mediates connections between the RBC membrane and the spectrin-actin cytoskeleton. So we have a lot of biochemistry drawn here in terms of knowing some of the components. So we know that band-3 is a critical component of this complex, and this is a 97 kilodalton integral membrane protein. It's a dimeric anion exchanger, which exchanges chloride and bicarbonate across the membrane of the red blood cell. And while it's typically expressed as a dimer, it appears to form a tetramer in the context of the ankyrin complex. We'll come back to that in a minute in terms of whether it's actually a tetramer or maybe something else. Then we have protein 4.2, the role of which has been kind of unclear, although it's known to regulate the association of band-3 and ankyrin. Ankyrin itself forms the core of this complex. So this is a 200 kilodalton scaffolding protein, which mediates the association of membrane proteins to the spectrin-actin cytoskeleton. And then we have Glycophorin A, which is a single transmembrane protein that is known to regulate band-3 expression and localization to the plasma membrane. On its own, Glycophorin A is known to form a dimer, a dimer of two single transmembrane segments. And you can see over here a schematic representation of what this is believed to look like in broad terms. Okay, so one of the things that I mentioned was that this complex is responsible in large part for the biconcave disc shape of the erythrocyte. So how do we know that? Well, we know that because mutations in basically all of the elements of this complex are linked to disease, and they're linked to a couple of different diseases, but most prominently hereditary spherocytosis, or HS. This is one of the most common inherited anemias in humans, And it's characterized by a complete disruption of that biconcave disc shape. So instead of having red blood cells, which are that classic biconcave disc, instead you have spherical red blood cells in HS patients, where the cells themselves are these kind of irregular spheres that are very fragile and which are rapidly degraded by the spleen, leading to the anemia phenotype. And this is thought to be due to the fact that you've disrupted the ankyrin complex and therefore disrupted the linkage of that complex to the cytoskeleton, which is presumably somehow templating the shape of the red blood cell. Okay, so I've talked a little bit about the linkage to the cytoskeleton and how this is binding to spectrin. But the other side of this is that the membrane proteins which ankyrin is actually acting to bring together in the plasma membrane are functionally related. And it's been proposed in the past that the ankyrin-1 complex actually forms a carbon dioxide metabolon, which facilitates rapid gas exchange across the red blood cell membrane. And so you can sort of see why if you have a look at some of the proteins that are involved here. So, we have band 3, obviously, which is involved in exchange of chloride and bicarbonate. And bicarbonate itself can be exchanged by carbonic anhydrase, which binds to band 3, to gaseous carbon dioxide and water. So there's one link for you. And secondly, we also have potentially associated with this complex or potentially in another subcomplex, we have the Rhesus polypeptides, RhAG and RhCE, which are known to form an ammonia channel, which facilitates diffusion of NH3 and consequent, alteration of the pH and regulation of the pH within the red blood cell. And Rh itself, apart from being involved in ammonia transport, has been theorized to have a role as well in carbon dioxide transport itself. So bringing all these things together in the one place could allow for even more rapid diffusion of carbon dioxide across the membrane and exchange of carbon dioxide from the soluble to gaseous state than would otherwise occur. Okay. So what don't we know here? Well, we know some of the components, but I don't think we know all of them. And we certainly don't know the stoichiometry. We don't know how many of each component is associated in this complex, and we don't know how they fit together in three-dimensional space. And we don't know how ankyrin binds the membrane. We've never seen ankyrin binding to an integral membrane protein before. We've seen it binding to peptides from integral membrane proteins, but we've never seen it binding to a actual integral membrane protein in the membrane or in a detergent micelle or in a nanosk. And we also don't know how this complex is involved in modulating membrane curvature. So our goal was to structurally characterize this complex from the red blood cell. So normally, if we were approaching a structural biology project, we would try to reconstitute a complex recombinantly, express all the components in some system where we can get a nice homogeneous sample. In this case, that just wasn't possible because we don't necessarily know all the components and we don't know how many copies of each component we have. And we're fortunate in that this complex is one of the most abundant membrane protein complexes in erythrocytes. So we can take advantage of that in order to purify it. So how did we purify this complex? Or I shouldn't say we, Francesca Valese, an associate research scientist in my group and a phenomenal membrane protein biochemist and structural biologist, led this entire, pioneered this entire procedure. So first, Francesca solubilized the membranes using digitonin, and really identifying digitonin as the detergent of choice was a critical step. When we used other detergents like Triton X100, this complex completely fell apart into band 3, ankyrin, and the individual components. She then applied those solubilized ghost membranes to a glycerol gradient in order to separate out the high molecular weight complexes from free band 3 using density gradient centrifugation. And we then applied the high molecular weight fractions, so these guys over here, to a size exclusion column, so to a Super 06 column. And then another critical step here was the use of mass photometry in order, so basically a single molecule light scattering technique, in order to identify which gel filtration fractions had a well-defined high molecular weight complex present. So the main peak on gel filtration, which you can see here, we could see by mass photometry that that was about 400 kilodaltons. And given the size of a digitonin micelle and the size of a dimer of band 3, we didn't think that was sufficient for a bona fide ankyrin complex of band 3. Having a look at this little shoulder that you can see over here, we can see that this actually has a well-defined complex of about 1200 kilodaltons, about 1.2 megadaltons. And, you know, this is kind of retrospective, but we could see there was enough room here to fit a lot more than just band 3 alone. And you know, we hoped that this would correspond to an ankyrin complex, maybe not the ankyrin complex, but hopefully some subfraction of the endogenous ankyrin complexes in the erythrocyte. Okay, so we then applied that to a grid and collected a nice big data set on a Titan Krios, and you can see what the raw data looks like here. So this is a representative micrograph. You can see just looking at the micrograph that it is very heterogeneous, but we do have very well-defined particles, right? You can see that here we have a very large micelle. We have some stuff outside the membrane. We also have some top views of the same complex, another side view over here, another different side view over here. And when we perform 2D classification on that data set, we can see that it is indeed very heterogeneous. And we have quite a mixture of small and large species here. And I've highlighted in green the large species and in red the small species. And we'll get in a little bit to what those species actually represent. So the data processing workflow was, you know, quite complex, and I'm not going to go through all of it in detail, but in a nutshell, you know, we first, well, not shown here, we did a first round of particle picking using a Gaussian based picker, blob picker in CryoSpark, 2D classification, which is what you saw in the previous slide. And then we use that to identify the micrographs that were the very best, the ones that had the most good particles on, and use those in order to train a topaz model to go back and repick the entire data set. From that, we obtained 1.8 million particles, and we went into heterogeneous refinement using models that we had identified from subsets of 2D data. So basically, we're using 2D classification here as more of a analysis tool and as a tool to identify homogeneous subsets, which we can then use to identify initial 3D models that we can put into a reference-based classification approach that we then apply to the entire set of topaz-picked particles. And that actually helped quite a lot in identifying, you know, well-defined smaller subfractions of data, right? I'm not going to go through all of the rest of it right now, but one thing that I wanted to just sidebar, which was maybe a little different. So we have some very flexible transmembrane regions here, notably one of the band 3 regions and one of the Aquaporin regions that I'll get to in a little bit, where the initial maps that we obtained by focused refinement were not too great. So in this particular case here, we obtained a map by initial focused refinement from the overall consensus reconstruction of about 4.5 angstroms, and really a very messy 4.5 angstroms, not something that I would want to build into. And applying focus classification to this did not yield good results. We couldn't use focus classification to get a clean set of particles that went to high resolution. So we took kind of a different track, which was to apply to use 3D variability analysis in CryoSpark with a mask around the region that we performed local refinement on. So we first did local refinement, then we did 3D VA, and then we clustered just on a single component that you can see here, which seemed visually to correspond to kind of an order-disorder transition. So, basically a transition between good and bad particles, for want of a better word, or junk and real particles. Localized junk, something that either didn't have band 3 or had disordered band 3. I don't exactly know, but clustering on that single component allowed us to identify a subset of particles that then went to much better resolution and had much better defined density, in this case for band 3, but we also applied the same approach to another component of the complex that I'll get to shortly. Okay, so just recapping there, a couple of highlights. So topaz picking really did make a big difference. And actually, I'll get a little bit later to this, but using topaz to repick with a model that was trained for a particular subset of data actually made a real difference for one of the complexes that I'm going to talk about in a second. However, I would just highlight that even a neural network-based picker like topaz still isn't as good as your own neural network, or at least in this case, it wasn't. So we compared, a couple of thousand manually picked particles to the equivalent micrographs picked by topaz, and the 2D classes are still a lot better from manually picked particles. Unfortunately, manual picking is not a very scalable solution for a 14,000 micrograph data set. But we've got room to make this better, I think. I just want to put that out there. Like I mentioned, we only really use 2D classification as an analysis tool to identify, okay, our data is good, and to particularly identify certain homogeneous subsets of data, which we can then plug into a stochastic gradient descent based ab initio reconstruction approach to generate initial models that we can then use for reference-based classification against the complete data set. Like I mentioned, 3D variability analysis is really helpful in order to identify subsets of particles which had homogeneous transmembrane regions for certain very flexible regions of the complex, in cases where focused classification didn't work for whatever reason. Okay, so I mentioned this is a heterogeneous data set. So here's a broad overview of that heterogeneity, just in broad terms. And what you can see is that we have 6 overall classes for the ankyrin complex. And then we have one separate class corresponding to those small particles, which appeared on initial inspection to correspond to free band 3 dimers. I'm not going to go into all of these classes in detail, but there's a couple that I want to highlight particularly. So the first one that I'm going to get into is class 1, which we believe to correspond to perhaps the most complete class that we have in this data set. I just want to highlight here, I'm not going to talk about it much, but there's still some unknown territory in this data set, which is one of the reasons why we want to get this data set out there on Empire. So class 4 here, we have a totally unknown component, which so far we have been unable to get to high resolution, but which I believe corresponds to a novel component, or maybe not novel, but at least a component that we have not yet identified to high resolution, which for the moment we're terming component X. And this corresponds to a rearrangement in the transmembrane domain of some of the other components. Okay, so what does class 1 look like overall? So I just want to show you a little movie here that will walk you through the overall architecture of the erythrocyte ankyrin complex. And this beautiful movie was prepared by Kuk-Ju Kim in my lab and Francesca Valese together. And so we'll start off just rotating around, and you can see that we have a very large region in the intracellular region. In red, we have ankyrin here. You can see snaking around in the core of the complex. and one particular part of ankyrin that is interacting with the RH complex. So you can see one of the big surprises here was that the RH complex is actually forming the core of the complex, interacting with the membrane binding domain of ankyrin. And surrounding that, we have 3 copies of the band 3 anion exchanger, which themselves are bound to Glycophorin A. And one of those copies, the interaction is mediated by protein 4.2. So protein 4.2 appears to be acting as an adapter. And I'm just going to show that one more time so you can get a better view of it. One of the things that you might notice here is you have this superhelical region of ankyrin, and part of that is interacting with protein 4.2, but the N-terminal region of ankyrin is actually not continuous with that ribbon shape. It's broken off and oriented orthogonally with the peptide binding groove directly facing the membrane. And that was a real surprise, which I'll get to in a second. And you can see here the heterotrimeric arrangement of the Rhesus polypeptide complex here. One of the other things you might appreciate is that the cytosolic region of band 3 is extremely flexible. I don't know whether you've noticed here, but one of these band 3 cytoplasmic domains is actually upside down with respect to the others, particularly the one that interacts with protein 4.2. Okay, let's move on. All right, so this is just a broad overview of the membrane association and target recruitment, basically, by ankyrin. So in the core of the complex, you have ankyrin here in red that is interacting. And I'm not showing here the transmembrane regions of band 3 because it's just too confusing, but they are there. I'm just showing the cytosolic regions that interact with ankyrin and protein 4.2. So you can see that first ankyrin directly binds to the cytosolic, termini of RhCE. So specifically RhCE, not RhAG, via the first 5 ankyrin repeats. Protein 4.2 has this kind of flat P-shaped arrangement that's oriented vertically and acting as an adapter with an extensive interface with the basically the second subdomain, for want of a better word, of ankyrin, while the other face of protein 4.2 is acting to recruit what we call band 3-1. We've named the 3 band 3s, band 3-1, band 3-2, and band 3-3. I apologize for our lack of imagination. And then we have band 3-2 over here interacting with sort of the first curve of the outer face of ankyrin, while the cytoplasmic domain of band 3-3 binds to sort of the very end of the ankyrin membrane binding domain. Notably, and not shown here, the N-terminal peptide of band 3-3 runs right back along the inner groove of ankyrin. And there's a very well-ordered N-terminal peptide of band 3-3 that sits in the peptide binding groove of ankyrin approximately here. Okay, so I've mentioned ankyrin, but I haven't really gone into much about it. Somebody is unmuted. If they could just mute themselves, that would be great. So there are 3 ankyrin proteins present. Okay, just wait a second. All right, so there are 3 ankyrin genes in vertebrates. We have ankyrin-1 or ankyrin R, which is the one that we have in erythrocytes, ankyrin-2 or ankyrin B, and ankyrin-3 or ankyrin G. If whoever's unmuted could just mute themselves, that would be great. So ankyrin share a common domain arrangement, which consists of a really characteristic 24 repeat membrane binding domain. So it's got repeats of 33 amino acids, which form this kind of superhelical arrangement, followed by a spectrum binding module and an autoinhibitory motif located in the C-terminal tail of ankyrin. Okay. Ankyrin, doesn't normally look like this, right? It doesn't normally have this T-shaped arrangement, or rather, we didn't know about this before. And what I mean by that is if you look at prior structures of ankyrin-2 of the ankyrin-2 membrane binding domain, what you can see is that even the 24 repeat structure of the ankyrin repeat membrane binding domain, it forms a continuous ribbon. The first 5 repeats are contiguous with the rest of the repeats in the structure. What we see when ankyrin is binding to the membrane is that this first 5 repeats are oriented orthogonally to the subsequent repeats, with just a little linker between repeat 5 and repeat 6. That itself, that linker is pretty well ordered. And this positions it such that the protein binding groove or the peptide binding groove of AR1 to 5 directly faces the membrane, which obviously sets it up to bind the membrane embedded targets, RhCE, and as we'll get to, Aquaporin. Okay, so you might wonder, like, is there something particular about this region of the ankyrin membrane binding domain? There kind of is. So if you take apart the sequence of ankyrin-1 and you just align all of the ankyrin repeats to one another, what you can see is that in most of these repeats, there are some pretty conserved regions, and the length of the repeats is almost absolutely conserved, with the exception of repeat 5 and 6, which is the interface that we see quite dramatically changing upon membrane binding. So you can see that repeat 5 is 4 amino acids shorter here at the C terminus of that repeat. And there are also some differences in regions that are otherwise very highly conserved. Is this the same in other ankyrin? Yes, it is. So if we take the 5-6 2 repeat module and compare that between ankyrin-1, ankyrin-2, and ankyrin-3, you can see that it is very well conserved, and that linker between the 2 repeats is absolutely conserved. And to us, this suggests that this dramatic rearrangement may also be conserved in the context of other ankyrin complexes that occur in neurons, in kidney epithelial cells, and other contexts where other ankyrin genes are also mediating clusters of membrane proteins. So like I mentioned, we have a band-3 N-terminal peptide that is sitting in this groove along the C-terminal membrane binding repeats of ankyrin. And it terminates right at the interface with that first subdomain of ankyrin. Interestingly, if you look at the structure that I mentioned of ankyrin-2, that structure is actually bound to an autoinhibitory peptide of ankyrin-1. And in this case, that autoinhibitory peptide traverses that interface, but it traverses that interface in a context where it's forming that continuous ribbon-like architecture that I mentioned earlier. So our thinking about this is that maybe that is part of the mechanism of autoinhibition. Basically that when ankyrin is in solution and in an autoinhibited conformation, it can't bind to the membrane because one, the peptide binding groove is occupied by the autoinhibitory motif, but also it's just geometrically not positioned to interact with the membrane. And when that autoinhibitory motif is substituted by the N-terminal peptide of band 3, I'm sorry, there's a typo here, this should say band 3-3, not band 3-1, that facilitates the conformational rearrangement that allows for membrane binding. That's a little speculative, but I think we have some circumstantial evidence of that. Okay, so here's just a little movie to show you in more detail the arrangement of that peptide. So you can see here the cytoplasmic domains of band 3-3 and the initial region extending back towards the N-terminus. We can actually see density for this intervening bit, but not good enough to build. And then here you can see this little 2-alpha helical segment that is bound in the peptide binding groove. with the corresponding density map, which you can see is pretty nice here. All of this region is very well defined at about 2.4, 2.5 angstroms. And you can see that the density more or less backs that up. So RhCE binds to the ankyrin-1, first 5 repeat region in the same direction as observed for the binding of the NAV1.2 N-terminal peptide. One of the things that I didn't mention here about this autoinhibitory motif is that's actually binding in the reverse orientation to what we see for the band-3 peptide. So they're binding to the same peptide binding groove, but in reverse orientations, which was kind of a surprising observation to us, at least. Okay, so I've mentioned the Rhesus complex or the RhAG complex, but I haven't really gone into what it is. So humans express 5 Rhesus polypeptides. In erythrocytes, we have RhAG, RhCE, and a very similar one to RhCE known as RhD. And then we have RhBG and RhCG in the kidney epithelium. And so previously, we knew that the Rhesus complex was likely to be a complex of RhAG and either RhCE, RhD, or both, but we did not know the stoichiometry. We expected it would be a trimer based on previous homotrimeric structures of, I believe it was RhCG, but we didn't know what the heterotrimer looked like or how it was put together. And we can now see that the Rhesus channel is a heterotrimer that's formed by 2 copies of RhAG and 1 copy of RhCE. And particularly RhCE is acting to form the primary attachment point for ankyrin, mediated by the cytosolic N and C termini of that protein. Okay, so moving back for a second to band 3. So like I mentioned when we saw that movie of the complex, the orientation of the band 3 cytoplasmic domains differs dramatically between the 3 different copies of band 3. And you can see that represented here. So these are all the same protein. The transmembrane regions are in gray and the cytosolic regions are colored by the different copy of this dimer. And so there's an extremely flexible and quite long linker between the transmembrane region and the cytosolic region of the protein. And it seems to take advantage of that by adopting very different orientations in the context of different epitopes that interact with ankyrin, and most dramatically in the context of the different region that interacts with protein 4.2. So I don't know if it's very easy to appreciate here, but the copy of the cytosolic domains of band 3.1 here that binds to protein 4.2 is completely inverted. It's upside down when compared to the 2 that interact with ankyrin. And that has implications if you think about potential interoligomeric interactions between different complexes of ankyrin, which is something that we don't know if that occurs, but it's something that we're certainly thinking about as a possibility. Okay, so what is protein 4.2 doing in all of this? So this structure also represents the first structure of protein 4.2. I should note that for model building here, I initially generated this model using TR Rosetta, and it was actually, very, very good. There were, some regions that were not perfect, but the overall domain architecture was spot on. And so we were able just to fit that into the density map and then do a bit of flexible fitting and basically get us there. And we went back and tried it with AlphaFold. AlphaFold in this case, for some reason, did not do quite as good a job as TR Rosetta, although it did give the overall fold, very well. I don't know why that is. We've tried AlphaFold on many other things and it's worked beautifully, but it's just an observation. So here we can see that the role of protein 4.2 seems to be to act as an adapter, stabilizing the interaction of ankyrin with the membrane and allowing the recruitment of this additional dimer of the band 3 anion exchanger. both via interactions with the cytoplasmic region, which you can see here, as well as with some involving some interactions with the transmembrane or juxtamembrane regions of the band 3 anion exchanger. I should note here as well that the N-terminal region of band 3 also plays a significant role in its association with protein 4.2, just like it does with the association of band 3 with ankyrin. And this makes sense because we know that if you remove the first few amino acids of band 3, you don't compromise membrane trafficking of band 3, but you do compromise the formation of the ankyrin complex. And now we can see why that might be. Okay, so just to go back and briefly look at one other interesting complex here, we could see in one class that we had something here that looked a little bit different, kind of square-shaped. And from the density, we were able to identify that as Aquaporin I, so as the water channel, Aquaporin I, which was quite unexpected, I would say. We had some indications from the literature that Aquaporin I might be at least functionally associated with this complex, but we did not know that it was a bona fide member of the ankyrin complex until we saw it in the density map. So just to give you a tour of this complex, so this is a sub-complex here where we have one copy of the RH complex, I'm just showing the density map and the model. One copy of band 3, so band 3.1, the same as in the other one that I showed you, as well as ankyrin-1 and protein 4.2. And by doing a mask refinement of Aquaporin I, recentering and applying C4 symmetry, we can actually improve the density here to get a nice 2.8 angstrom reconstruction of Aquaporin I in the context of the complex, which pretty unambiguously identifies this as Aquaporin I. Okay, so I just showing that again in case you didn't see it very well the first time, and also because I just like pictures of pretty density maps. You'll also notice there are some cholesterol molecules here. Well, cholesterol molecules or digitonin is, potentially a matter of interpretation. I think they are cholesterol, but it can be hard to tell the difference sometimes. Okay, so what does this look like here? So we can see that Aquaporin I is located at the mutual interface of band 3.1, protein 4.2, and the very end terminus of that 5 repeat segment of ankyrin. And so the interaction between Aquaporin I and these proteins seems to be mediated by the interactions of the N terminus of Aquaporin I with ankyrin, as well as interactions of the C terminus of Aquaporin I with domain 1 of protein 4.2, which you can see over here. Okay, so just taking it back a step and revisiting the carbon dioxide metabolon. So I mentioned that, the ankyrin-1 complex was thought to form a carbon dioxide metabolon. Well, now we can add another little potential wrinkle to this because we know that Aquaporin I conducts both carbon dioxide and water. And you can see over here that both of these are substrates for carbonic anhydrase, which we also believe, although we have yet to structurally see it, is associated with this complex. So it makes sense that Aquaporin I, being in this picture could be helpful for facilitating, rapid carbon dioxide exchange across the membrane. Okay, so just going back a second and looking at one more of these complexes, one more class in this heterogeneous mix, which we believed was free band 3. So we thought it was free band 3, but when we actually solved the structure and we were able to initially obtain a 2.8 angstrom reconstruction of this particle, essentially as a contaminant from our initial round of picking. So we obtained a 2.8 angstrom reconstruction from 37,000 particles. And we could see that there was an extra transmembrane helix in addition to the ones that we expected for band 3. And we found that that was actually Glycophorin A, which is a single transmembrane protein that's known to regulate band 3. Interestingly, Glycophorin A is classically a dimer, and on its own, it is a dimer, but it binds as a monomer to band 3. We don't know why that is yet, but what we can see is that comparing the structure of the isolated GPA dimer to what we see in the complex, we can see that the interhelical angles in the dimer, which are quite pronounced, would be incompatible with band 3 binding. So basically, it must come apart in order to bind to band 3. We don't quite know how that process works yet. One other interesting observation here was that we saw a very well-ordered lipid, and we believe that that lipid is actually PIP2 based on the density. We don't have any other evidence for this, but the density has a very characteristic shape with these two apparent, phosphate groups and this, sugar, and it's binding in a very positively charged pocket that is surrounded by lysines, arginines, and so on. So we believe this is PIP2, which is really interesting because generally speaking, PIP2 is a signaling lipid, and it's a lipid that's only present in minor proportions in the cell membrane. So usually if it's there for a reason. Whether that reason is structural or functional, we don't know, but it's a subject of active further research. Okay. So I mentioned
Oliver,
that
sorry to interrupt you. We're about to running over time, so maybe if you could wrap up quickly.
I've got a couple. Yeah, I've only got a couple minutes left. Is that okay?
Yeah, cool. Thanks. Yeah.
Okay, great. So I mentioned that this structure we initially obtained at 2.8 angstroms. So actually over the weekend, we were able to improve that to 2.5 angstroms. So this is brand new. And so what we did there was we used topaz, but we used topaz a second time. So we could see that many small particles were being missed in micrographs that were packed with big ankyrin complexes. And so what I did was I sorted the micrographs based on the number of band 3 GPA particles that were remaining, identifying those that only had the most small particles that were contributing to that 2.8 angstrom reconstruction. And then we kept only the very best, the ones with the most particles, the ones with the thinnest ice. Curated those picks, added a few extra ones manually, trained a new topaz model, and then repicked the entire data set. This gave a really big improvement. So it allowed us to go from 37,000 particles and 2.8 angstroms to 266,000 particles and 2.5 angstroms. And I hope we will be able to improve that somewhat further. And just to give you a picture of what that looks like, here's a representative section of that map and the associated atomic model. And you can see that the density backs that up. We have very well-defined carbonyls and very well-defined rotameric states of small hydrophobic side chains. Okay, so I mentioned this class 1 and this class 2. We wanted to see, is it possible that are these actually compatible? Could these actually be part of the same complex in vivo? And just by aligning them together and having a look at it, eyeballing it, you can see that certainly, sterically they're compatible. It's entirely possible that Aquaporin I forms part of the native complex in conjunction with 3 copies of band 3. We can't prove that yet, but we're getting some ways towards doing that. And in order to do that, we're moving to cryo-electron tomography. So Francesca made a beautiful preparation of native red blood cell vesicles by extruding red blood cell membranes and then applying them to a grid, blotting to get nice thin ice. And then we screened those on a glacios, and we could see that we had very well-defined vesicles that are decorated apparently on both the inside and in some cases the outside with protein complexes. We then established a collaboration with Alex Noble's group down at the Simon Electron Microscopy Center to start to do some cryo-ET. And you can see one of these tomograms here collected on the Krios down at SEMSI, and you can see that there are really nicely defined protein complexes decorating these vesicles, as well as some membrane fragments. And if we have a closer look, you can see that the shape of these complexes seems pretty characteristic and very reminiscent of what we see from our single particle data. Okay, just skipping through a little bit. So we actually did very recently, and this is not yet in the preprint, but we did do some sub-tomogram averaging, and we're able to obtain a nominally 30 angstrom, although to my eye it looks a little better than that, reconstruction of membrane-bound endogenous ankyrin complexes. And they look very, very similar to what we see from our single particle work. Sorry, this movie just seems to be glitching a little bit, but hopefully as you rotate it around, you can see we've fit our atomic model in here, and it matches up pretty well with the map. And you can see a cut through of the transmembrane region here. And if you got a bit of imaginitis, you can see that there might be some density for the RH complex and maybe for some of the other transmembrane proteins. But we hope to improve this further in the not too distant future. Okay, so I'm going to skip through the conclusions because I think they're pretty obvious at this point and we're running out of time. The most important thing is to thank and highlight the team who are involved in this work. So this is Francesca, who really was the hero here, who led this entire project and took on a really challenging project in the middle of the COVID pandemic and took it to a successful completion in a very short amount of time. We started this project last March, more or less, and collected the first Krios data in July. And now we have what I'm really quite proud of as a nice story, and that is all credit to Francesca. This is Zhening, who helped us out a lot with data collection, Laura Yen, who was rotating in the lab and helped out with purification, and Gladwin Gillis, who was a Spurs student in our lab at the time. And this is a photo we took during the data collection, which ultimately led to the structure. As I mentioned, just thanking Francesca, Kukju Kim, who made a lot of the figures in this presentation and helped out an enormous amount with data analysis, Laura and Gladwin, the Mancha Lab, everybody in the Mancha Lab was a phenomenal support and contributed a lot in terms of critical reading and just general support. The Cryo-EM Center, led by Bob Grisucci and Zhening Zhang at Columbia, has been fantastic. And we've had a fabulous collaboration with Alex and Jake at SEMSI. And Tito Kali's group did an enormous amount of work initially in optimizing the preparation of the ghost membranes that we used as raw material to purify the complex. And that's all I've got for you. Thank you very much for listening, and I'm more than happy to take questions. I do apologize for running a little bit over time. I'll just stop sharing now.
All right, thanks, Oliver. Great talk. So now it's question time. So I see Peter raise your hand here. Peter, do you mind to unmute yourself and ask your question, please? Thanks.
Hey, Ollie, amazing work as usual. Nice to see you still doing cool stuff. The hereditary spherocytosis mutations, you've obviously looked at those and how they place. So do they place on the spectrin side or on these complex sides, on the ankyrin?
So they're, yeah, great to see you, Peter. And yeah, I hope I'll be able to see you in person sometime soon and make it back. Just a few visa issues to sort out. But yeah, they're actually all over the place. So the most common HS mutations are roughly in order of appearance: spectrin, ankyrin, band 3. There are a couple in RH, although not that many. And then there are also, I believe there might be some in Glycophorin A, but I'd need to double check that. And most of them just completely disrupt the complex. So they knock out expression of band 3, or totally knock out the architecture of the cytoskeleton, more or less. There are some which don't. So the one exemption to that is there's what's called the Neapolis mutation, which corresponds to a deletion of the first 12 amino acids of band 3. And in that case, you still get quite a lot of band 3 making it to the membrane. And that's one that we would be quite interested in looking at, structurally, to see what's going on there.
So what's the consequence of that in other ankyrin complexes in other cells, for example, in neurons? It seems really
That's a very good question. Yeah, so there's actually some overlap in terms of like mutations in spectrin also have a neurological phenotype in some cases. And yes, it is quite important. We're not there yet, but we're definitely interested in having a look at some of the other ankyrin complexes, because my suspicion is that, like, it's not going to look the same, right? But I'm betting that there's going to be something that is acting like RH is acting and is interacting with those first, 5 repeats of ankyrin. And there will be other things that are acting like band 3 is acting, and acting as kind of the satellites.
Awesome. Cool. Thanks, Ollie.
Thanks, Pete.
Cool. Thanks. And we have Dongju raise your hand now. Dongju, do you mind to unmute yourself and ask the question?
Hi, Oliver. It was a really great talk. My question is the band 3 conformation affected by different pH level, because it works as a anion exchanger?
Yeah, that's a really good question. So actually, there was a lot of early work on, but before there was a structure of the band 3 transmembrane domain, which there's a structure of that going back to about 2015 in the outward facing conformation. There was a lot of work on looking at exactly that, and it appeared that there's come some kind of structural transition in the cytoplasmic domain when you go from low to high pH. We haven't really looked at that at all in the context of the intact complex, but I think it would be really interesting to do so, because, as you say, it makes functional sense for this to be regulated by pH.
Then the higher pH affect the whole conformation of the cluster?
So I don't want to get too far out of my skis, because I don't remember all of the details. My recollection is that you go from sort of an expanded to contracted state of the isolated cytoplasmic domain when you change pH, but I don't know what that looks like in the context of the full length protein yet.
Okay, thank you.
Thanks.
Rightio. So we're at 11 now. For those of you who have to leave at 11 o'clock, thank you for attending this seminar, and thank you, Oliver, for your presentation. That's really great. And please keep in mind that this recording will be made available on the CCMMP website. So yeah, have you got any more questions going on?
I have a quick one if we've got time.
Yes.
Sorry, Isabel.
No, go ahead, Patrick. I go.
I'm happy to hang out and chat. I got nothing to do this evening. So,
in terms of the cryo tomography that you've started doing, do you get any indication that having more intact sort of architecture of the membrane and so forth is influencing any of the complex? I mean, I know your resolution is limited at this point. I mean, is the membrane curvature the same, or is it different?
Yeah.
Or?
So that's a really good question. We're only really in the early days of that work. What I can say is that in that sample, we have both inside in and inside out vesicles, which may actually be an advantage, because if you think about a red blood cell, you have areas of the red blood cell that have both positive and negative curvature. So we're really interested in looking at what the complex looks like in both of those contexts. Right now, we don't have any direct, like looking at the reconstruction we have of the ankyrin complex from the sub-tomogram averages, it looks very much like class 1. Looks very much like class 1, and I believe that there is Aquaporin there as well, based on just inspection of the density map. But we gotta, I can be convinced, but we gotta get to a bit higher resolution before anybody else will be. The question of how it modulates membrane curvature, I'm not sure. One of the things that I've been thinking about, though, is that if you think about it, this complex has 3 band 3 dimers, and they are dimers. And that means that you have exposed ankyrin and protein 4.2 binding sites all around this complex. So my suspicion is that it may be to do with interoligomeric interactions of multiple copies of this complex. And if you sort of do the math, there are only a few different ways that that can work. And if you look at them, some of them seem to correspond to having some reasonable level of curvature there, but it's totally speculative at this stage.
Yeah, well, hopefully the tomograms will help answer these.
I hope so too.
Thanks, Oli, for a beautiful talk. I don't think we had the chance. You left WeHi before I arrived at WeHi. But actually, to follow up on Patrick's question, do you see, is it evenly distributed on the membrane, or do you see some sort of cluster? And can you relate this with clustering of other intracellular protein that will keep things together and actually probably in fact affect the curvature of the, you know what I mean? Is it?
Yeah, it's a, yeah. So in terms of inspection of the tomograms, to my eye, it definitely looks like we have multiple copies of this complex that are in one place. We don't have the, and doing a reconstruction in a slightly larger box size, you do see ghosts of what seems to be another copy of this complex appearing. But that's all talking about copies of the same complex. In terms of like interactions with spectrin and interactions with other intracellular proteins, I can't, you know, can't unambiguously say anything about that yet. The other aspect of that we would be really interested in looking at is trying to see if we can reconstitute a complex with carbonic anhydrase and see how carbonic anhydrase is tethered to the membrane, because I think that could be really interesting to look at.
And the quick other one is I'm quite puzzled, you know, the protein 4-2 binding to only one bound 3, even so there's 3 copies of it, but it's only one interface. So I suppose you dig out the specificity for this and explaining why it only binds to one. You know, well, I mean, it technically could bind everywhere unless there's another protein in the way for the other bound 3.
Well, you mean why doesn't protein 4.2 associate with the ones that are bound to ankyrin?
Yeah.
So the reason for that is actually that the band 3 cytoplasmic domain that interacts with protein 4.2 is upside down.
Okay. Right.
So it can only interact with, so therefore any band 3 cytoplasmic domain that's interacting with protein 4.2 can't then interact with ankyrin, and vice versa. We don't really know what that means yet or why that's the case, but yeah.
Thanks. Absolutely great talk.
Thank you. Nice to meet you.
Rightio. Do we have any more questions? Anyone in the chat? Let's click up here. No. Cool. So yeah, thanks again for joining us today, Oliver. It's a great talk.
Well, thank you very much, Hai-Chen, and thanks very much for having me here. It was a real pleasure.
Yep, cool. So yeah, that's the end of the meeting. Right.